Microbiology, most common in hospital autopsies than forensic autopsies. Swab from surfaces. Take a swab of samples from required site, meninges, intestine, bladder. Swabs from a superficial wound, mucocutaneous orifices, avoid edges. Introduce deep into the cavity. Procedure. Introduce tip of a swab into the area of interest and rapidly close the swab, stick in the holder after the swab has been inserted. From solid organ, microbiological samples from solid tissues, example spleen, surface of the organ is seared using a flat-faced soldering iron or a scalpel blade, which is heated red hot in a flame. Bunsen burner is useful, but if not available, soak cotton in spirit and use of a lamp. Figure 5.23. Take samples for microbiology from a spleen. Heating knives red hot, sterilizing surface of the spleen with red hot knife, incising sterilized surface of spleen, introducing swab, stick in a spleen, making a smear for direct visualization, sending another swab for culture. Swab insert and replace into sheath. A glass lid may be made for direct visualization, can be used with a solid organ such as kidney, spleen, liver. Another alternative is to remove a portion of tissue about the size of a small die, 1 centimeter side. Send for a microbiology. Spleen is the best organ to sample for diagnosis of septicemia. Blood from a peripheral and is also useful. So, in septicemia, spleen is the best organ to sample for diagnosis of septicemia. Blood from a peripheral vein is also useful. Collection of microbiology blood for toxicology. Before autopsy, this is to be preserved because of lack of contamination, which invariably occurs an opening body. Perform a cardiac puncture by passing a sterile needle through the 5th and 6th intercostal space anteriorly and apply gentle pressure on the syringe plunger. Technique is similar to that of pericarditis paracentesis. Blood is collected in appropriate blood culture bottle and transported to the microbiology department. During autopsy, blood for culture must be collected before organs are disturbed. Open pericardial sac, sear anterior surface of right ventricle using same method was aspirate 10 ml of blood using sterile needle and syringe. Place in a sterile stoppers containers. Brain take swab as indicated above. Alternatively, open subarachnoid space with a sterile scalpel. Take samples from the space by a sterile swab. Make smears. Also send for culture. Virological examination. Ideal, a piece of appropriate sample is collected under sterile conditions. Sample frozen at minus 90 degrees Celsius in a liquid nitrogen flask. Liquid nitrogen flask at minus 90 degrees Celsius. Second best, preserve 50% glycerol in a buffered saline. Okay, thank you.